Hi everybody. In today's lesson, we're going to continue talking about the reproductive system. At the end of our last time together, we were discussing spermatogenesis and how that's connected to mitosis and the HPG axis. Today, I'd like to transition into talking about the female reproductive system. Specifically, today we're going to be talking about how the female produces gametes. That is, how she uses meiosis over the course of her life from before birth all the way through menopause to make fertile gametes. Okay, so to begin this discussion, let us look at some of the structures that are included within the female reproductive system, just like we did with the male reproductive system. And so first of all, let's talk about some external genitalia. First of all, the mons pubis is the fatty area covered with hair over the pubic symphysis. And essentially this is some padding so this bone is not damaged. The vulva itself, so the female external genitalia, forms what is called a urogenital triangle with the clitoris being the most anterior, the opening to the urethra, or the external urethral orifice, being more posterior. Next comes the vaginal orifice, and finally the anus here. Okay. Note that these various orifices and sexual structures are surrounded by skin folds. We're going to see here in just a moment that these skin folds actually develop in both the males and the females. It's just that at particular times during the development, so that is during the second trimester particularly, is when more testosterone versus more estrogen is going to drive the development of these structures in different directions. Okay, so the female hormone, so estrogen, is going to drive the development of these structures to become two pairs of what are called labia or lips. The labia majus or majora are the more superficial structures and covered with hair. The labia minus or minora are immediately bordering the urethral orifice, vaginal orifice, and the clitoris here. The clitoris itself is anterior to what is called the vestibule, right? So the urethral and vaginal orifices. The clitoris has a structure that is actually very similar to the penis because, again, these structures develop both in males and in females. It's just at different, or at the second trimester, the estrogen is going to drive the development of a clitoris, and testosterone drive the development in the direction of a penis. And so there are similar structures in the clitoris. It is also an erectile body, right? So during sexual arousal, the clitoris will also become engorged with blood and therefore become erect as well. There is a glans of the clitoris, so the head or the exposed portion. And just like the foreskin or the prepuce of the penis is skin that hoods the glans, the same exact thing is occurring within the clitoris. And again, when I say that this is homologous to the penis, I mean that these structures have literally developed from the same exact tissues over the course of fetal development. What we can see here is two different pairs of images. Here is fetal genitalia at 10 weeks. Here is fetal genitalia at 12 weeks of development. Okay, so what we can see here are color-coded structures, right? So the urethral groove. So essentially this is going to be forming the opening to the urethra. Okay, and so those structures are in purple. And then surrounding those structures, we have blue, which is extra skin. And in males, this extra skin is going to become both the scrotal sac as well as the shaft of the penis itself. In females, these more external folds of skin here are going to become the labia majora. Okay, and the innermost folds that would actually become kind of the inner portion, inner tissue of the penis, become the labia minora. And so again, I just want to point out that these structures derive from the same exact tissues. It's just that different levels of different hormones at exactly the right time, right? So between 10 weeks and 12 weeks post-development is going to start driving the development in different directions. Okay, and I gave you guys a couple different links here if you're interested and exploring more about this topic. As I've said before, the difference between male and female is really not as large as what our society might imply that it is. Physically, the difference between testosterone and estrogen is because of the action of one single enzyme, and it is one hydrogen different from each other. Okay, so male and female hormones are incredibly similar to one another. Also, we share the same developmental pathway up to a certain point, and there's no hard and fast cutoff for, testosterone versus estrogen. There is, as I've said before, kind of a mixture, a balance of the two hormones and other hormones on top of that, which is going to derive the development of these structures. And so if there is, estrogenic compounds in mom's blood, they might actually cross over the placenta, and they might shift the development of both male and female baby to be somewhere in between. There are also a series of developmental abnormalities which are more common than you might expect. And so that's what this lecture is down here. So different disorders of sexual development, many of them linked to the presence of endocrine disrupting compounds within mom during fetal development. I also, this animation here, I feel like is very well done. It is showing the procedures involved in a sex change operation. And so I add this link for this lesson, not because it's exam material or anything, but because I personally think it's interesting that these surgeries are literally following kind of the same patterns and reusing the same tissues to change developed sex organs into sex organs of the opposite sex. And so they literally use the same homologous structures to just kind of rewind the clock a little bit. Right. So again, those videos are certainly not required of you, but just something a little bit interesting as a supplement. Okay. Okay, so those are the external structures. The female also has a series of internal structures that are used by the reproductive system as well as the endocrine system. Okay. Here we can see the vaginal orifice, again, separate from the urethral orifice. The male reproductive system uses the urethra for both reproductive and urinary purposes, but in females, these two are separate entirely. Okay. So the vaginal orifice opens up into the vagina or the birth canal itself. This is about a 4-inch long muscular tube lined with the same kind of tissue that is lining pretty much any structure that opens up to the outside world, your oral cavity, your nasal cavity, your urethral orifice, right? All of these things that open up to the outside world are, of course, lined with this cheaply made many, many layers of cells tissue. What is different about the vaginal canal, however, is that even though it is generally covered with mucus, it is not non-keratinized. That is, it has keratin, which is the same substance that's in your more superficial skin. And so this is just an extra layer of protection against abrasion during sexual intercourse. The hymen is a vascularized membrane present in young girls. This can be broken not only by the first sexual intercourse, but also by other activities that the young girl might do. Okay, on either side of the vaginal orifice are something called the greater vestibular glands, or the Bertolin's glands. These are responsible for secreting fluids into the vaginal canal during the beginning of sexual arousal, not only to act as a lubricant, but also to interact with the sperm within the semen in some ways that are pro-sperm and other ways that are anti-sperm success. Okay. Again, these structures all have homologs. That is, they have structures that are similar in males and have all derived from the same structures developmentally. These are similar and homologous to the bulbourethral glands in males. So we can even see the Bertolin's glands right here. And so those are in pretty much the same place as the bulbourethral glands in males. Okay. Note that these structures are, of course, open to the outside world. And just like in the digestive system, right, so the digestive system being posterior here, the vaginal canal is populated by a community of microorganisms. And in a lot of ways, these microorganisms are helpful to the vaginal canal. They are going to maintain the environment. They are going to maintain the tissues they're in. Also, they fight bad microorganisms. That is, certain bacteria such as certain bacteria such as lactobacillus also colonize the vaginal canal, and therefore they secrete hydrogen peroxide into the local environment that actually suppresses a lot of the other things that would be detrimental to the female. And so one of those organisms that does kind of get, reduced by the good bacteria in the vaginal environment is yeast. And so the main yeast, which is a type of unicellular fungus, is Candida albicans or C. albicans. And so when the female's microbiome, right, both in her digestive tract as well as in her vaginal canal, are healthy, the yeast is suppressed significantly. Okay, and so when the female takes antibiotics or a lot of other pharmaceuticals that impair the health of her natural microbiome, that suppression by things like lactobacillus bacteria is not going to be enough to suppress the yeast. And so that's why yeast infections are fairly common after taking medications such as antibiotics. Okay. So again, Candida albicans is a naturally occurring fungus, but usually it is greatly reduced because of a healthy and diverse community of bacteria which naturally occur in the vaginal canal. Okay. The functions of the vaginal canal are both copulation, so reproduction itself, delivery of sperm to an area closer to egg, as well as childbirth itself. Okay. The vaginal canal opens into the uterus. So the uterus is a hollow, thick-walled muscular organ that receives an oocyte. Right? So a gamete, which we're going to get into here shortly. Also, it can retain and nourish this oocyte if it is actually fertilized into a true egg. Okay. Note that the uterus is usually portrayed like this. So in your high school sex ed classes, you probably saw the uterus like this, and maybe even at the doctor's office, you see the uterus kind of spread out like this. But I do want to point out that in reality, the uterus is what is called anteverted. Note that it is sitting pretty much right on top of the urinary bladder. And so this is why pregnant women have to pee so often, right? Because baby is literally sitting right on top of her urinary bladder. Also, there's an extra organ in here. So the uterus is, of course, not in males. And so the male bladder can be larger to fill in the space that is taken up by the uterus in females, right? So it's not her fault if she has to urinate more frequently. She doesn't have as much space as males do. Okay. So anteverted, it is pointing anteriorly. Note that in order from anterior to posterior, it is urinary system, reproductive system, digestive system in the female. Okay. Again, in males, the urinary and reproductive structures kind of share a couple structures instead. Okay. All right, so the body of the uterus is this kind of funnel-shaped portion here, and just like the stomach, there is a dome at the superior aspect of this organ, and that is called the fundus as well. Okay. The opening or neck between the vaginal canal and the uterus is called the cervix. And so you might remember that the vertebrae in your neck are called the cervical vertebrae. This is the cervical region. Well, cervix just means neck. And so this is yet another neck, another cervical region that females have. Okay. Surrounding the cervix here are a series of cervical glands, and these glands are tightly controlled by different hormones that are circulating within the female's body. So at different points during her monthly cycle, menstrual cycle, as well as uterine cycle, as well as ovarian cycle, we'll get into that here soon. But essentially, these cervical glands can secrete mucus that either promotes sperm entry or prohibits sperm entry. And so the human body was, of course, designed to promote fertilization, right? So in order to continue your species, it would make a lot of sense if your body was pro-reproduction. And so the cervical glands secrete mucus that promotes sperm entry during the middle of the cycle when the female is most fertile, and it's going to prevent sperm entry when she is not fertile. Okay. And so if you think about it, the entry of sperm when she's not fertile would just be kind of not helpful, not productive in terms of reproduction, in terms of evolution. And so her body would have to eliminate all of these extra contents, the sperm and the semen. And of course, there's no possibility of fertilization with it being present. And so her cervical mucus is going to inhibit the entrance of those fluids during a time when she can't possibly be fertilized. The uterine wall itself, just like so many of the other organs we've taken a look at, is composed of 3 different layers. Around the outside, so around the perimeter, is the perimetrium. Note that the uterus is retroperitoneal. Okay, so it is not actually covered entirely by the peritoneum membrane, but the peritoneum is actually kind of sitting right on top here, covering the intestines, which would be up here. Okay, the thickest portion is called the myometrium. Again, very muscular. Of course, the uterus has to contract in order to expel an infant, right, fetus to infant. But also it has to contract every month in order to dislodge the endometrium, right, the innermost layer here, to shed during menstruation. Okay, so again, this is the myometrium. Myo always means muscle. Endometrium is the lining of the uterus, which can be shed every month during a menstrual period. And also it can grow substantially if there is a fertilized egg within the uterus. Okay. The ovaries themselves, right? These are the primary sex organs. That is, they are the ones that make the gametes. They also make a lot of hormones as well. So these are the primary sex organs as opposed to secondary sex organs such as the breasts are secondary sex organs. They facilitate reproduction or child rearing, of course. Also, they are used as kind of a symbol of femininity, just like a beard in a man signifies that he has male characteristics. He makes sperm. Breasts, as well as a softer jawline, softer features, smaller muscles, all of these things are signifying at a glance that an individual essentially makes oocytes or makes eggs instead of sperm. Okay. So these ovaries, there are 2. Okay. The ovaries have 3 main functions. First of all, they are going to produce these immature female gametes. Note that these are called oocytes. Right, so the O's should remind you a little bit of eggs and nice round eggs. So oocytes are the immature gametes. And the process that actually creates these immature gametes, these oocytes, is called oogenesis. Okay, just like spermatogenesis generates sperm, oogenesis generates oocytes. Okay. So this process is actually going to occur within the cortex of the ovary. So yet again, we see this term that indicates the outside of an organ, the adrenal cortex, the cerebral cortex, the renal cortex. All of these different organs have an outer layer of the organ and an inner layer of the organ, so the medulla of the organ. And so if we take a look at this section through the ovary here, what we can see is the outer portion, which is filled with all these funny little circle things, as well as this like cotton ball cloud looking thing over here. These are developing oocytes within something called follicles, and we'll get into that here very soon. Okay, so these ovarian follicles are the nurturing structures that are going to both facilitate and regulate the meiosis of the developing egg, okay, as well as sustain a pregnancy for the first few weeks to few months, if that has occurred. And so these follicles are just, a series of cells that are really important in making the eggs and sustaining the eggs, especially if they are fertilized. Okay, and so we are going to actually break down the oogenesis into what is happening with the eggs and what is happening with the follicles. Both are critically important to understand in order to understand the female reproductive system. Okay. The ovaries, just like the testes, are also really important in making hormones. Okay. These organs are going to secrete female sex hormones. We are going to be talking most about estrogens and progesterone. Note that progesterone is pro, right? It promotes gestation, and gestation is just a fancy word for developing your offspring, your zygote into fetus, etc. So progestation, it is pro-pregnancy, right? So that should also indicate to you that it is pro maintaining the endometrial lining of the uterus. It is pro retaining nutrients for a developing embryo, okay, et cetera, et cetera. So anything that is related to pregnancy, progesterone is going to ramp up. Okay. Also, again, just like in the male reproductive system, the primary sex organs of the female are going to secrete a hormone called inhibin that literally inhibits the production of FSH, because once again, this entire system is going to be regulated by the HPG axis. Only this time, the G, the gonad part, is actually the ovary as opposed to the testis. Okay, so let us talk about oogenesis. We're going to begin our discussion by focusing on the egg itself. So on the oocyte, remember that we are going to be talking about what's happening to the egg, what's happening to the follicle, and then we're even going to add on what's happening in the uterus. Okay, so let's start with the egg first. We know that making haploid gametes, so cells that have one copy of DNA that are designed to join with a gamete of the opposite sex and therefore make a new genetically unique offspring. Okay. Oogenesis, just like spermatogenesis, involves stem cells. Okay, and these stem cells are genetically identical to all of the other cells within the female's body. These stem cells undergo mitosis to make more of themselves, and then the type B cells essentially are going to be renamed the primary oocytes. These oocytes are going to complete meiosis 1 and maybe meiosis 2. And so unlike in the male reproductive system, this generation of gametes is going to begin before she's even born. And so I really want to emphasize that you, right, the part of your genome, the half that came from your mom, started to be developed in your grandmother. Okay, so again, the creation of eggs, right, which ultimately go on to become you, begins when grandma is still pregnant with mom. And so then the process doesn't complete until fertilization occurs. Okay, so you really do go back all the way to your grandma. Okay, so this begins before birth. Here we can see a chart that we're going to be walking through, mitosis of the oogonium, or oogonia for plural, of these stem cells produces lots of these diploid primary oocytes. Okay, the stem cells themselves, again, are called oogonia, right? And the daughter cells of these stem cells can become primary oocytes. And again, during mom's fetal development, the process of meiosis 1 is going to begin. And so this is going to happen a little bit differently than in males. We still have the whole crossing over portion, so synapses and mixing of those genes from grandma and grandpa. We still have the lining up of the metaphase plate. We still have all those things that happen in a male. But in females, prophase 1 is going to begin. Okay, so here we can see the copies of DNA. Okay, but then it stops. Okay, so it begins, but then pauses in the very first stage of meiosis 1. Okay, and so these cells, these primary oocytes, are going to be arrested within this phase. That is, they're going to be frozen in time. Someone hit the pause button until 2 weeks leading up to ovulation. All right, so this may be 12 years later. This may be 40 years later. Okay, so those very same cells began this process before mom was born, and their process doesn't complete itself in reality until fertilization occurs. Okay, while meiosis is happening in these cells, there are a series of cells that are going to surround this developing cell, right? This meiotically active cell. And so that kind of shell around the primary oocyte is called a primordial follicle. And so primordial should make you think of something ancient. And so the idea here is that these are the oldest of all of the follicles. Okay, so, when mom is 40 years old and she is about to ovulate this particular egg that started meiosis before she was born, that primordial follicle, that 40-year-old follicle, is going to start changing. Okay, so primordial is the oldest of all, is the first follicles that we see. Okay, so when mom is born, her ovaries are supplied with a lifetime supply of oocytes. Okay, so she does not ever begin meiosis after birth. Right, so as soon as she's born, that is as many oocytes as she is ever going to have. At birth, there are about 2 million living cells. Okay, 2 million living oocytes. Only about 400,000 of these remain by the time she reaches puberty. Okay, and so these numbers continue to go down over the course of her life. And of course, you can think about, how many ovulations, how many menstrual periods a woman has over the course of her life. And really, she's only ovulating a couple hundred eggs. So 400,000 is enough, right? Compare this, though, to males who are producing at least 15 million sperm cells at a time. Okay. Okay, so again, we're going to walk through this image right here. We've begun our discussion with talking about the stem cells, making more of themselves. Then these primary oocytes, again, still within the ovaries, are going to start dividing or start the process of meiosis 1, but they are arrested in prophase 1, literally the first step of this process. They're going to hang out there until she reaches puberty. Every month, in theory, from when she reaches puberty until she passes through menopause or a few of these primary oocytes, again, surrounded by their primordial follicles, are going to be activated. And we still don't have a good idea, as far as I know, exactly why certain oocytes are activated and why others are not. Okay, so jury's still out, more research to come, I'm sure. Okay, so of these few primary oocytes that are activated, one and only one, in theory, is going to be selected each month to resume meiosis 1. Okay, and again, we don't really understand why certain ones are activated selected and why other ones aren't. Of course, if you were on fertility treatments, this may increase the number of activated and selected oocytes every month. Also, in the case of twins, for whatever reason, multiple eggs are sometimes activated, resume meiosis 1 and ovulated, and so in theory, both of them can be fertilized. Okay, so one is selected to resume meiosis 1 each month. Okay, so here's that primary oocyte that's been selected. It is going to complete the process of meiosis 1, and so that's what this little process is right here, essentially pulling the two copies of DNA into different cells. Now, another difference between spermatogenesis and oogenesis is that this cell division is not equal. Right, so spermatogenesis is going to produce 4 equally sized, equally capable gametes. Here, in females, this process is shooting to produce 1 gamete and only 1. However, meiosis is going to produce 4 individual copies of DNA. And so pretty much meiosis occurs in females. In order to kind of get that DNA out of the way, we need to just take 1 copy and then another away so that 1 single egg, ultimately down here, can be ovulated and potentially fertilized. Okay, so where I'm going with this is that meiosis 1 is completed when cytokinesis, so true cell division, occurs, producing a much larger cell called the secondary oocyte and something called a first polar body. And so the first polar body is really just there to store the extra DNA that we don't need, we don't want. It's just superfluous at this point. And all of the other cytoplasmic contents, all of the other organelles and energy and sugar and everything are going to remain in the secondary oocyte. Okay, so essentially, the secondary oocyte is taking the stuff, the contents of 4 gametes, and packing them into 1 gamete. Okay, so when you look at images of sperm and egg, the oocytes are always huge because the oocytes contain cytoplasmic contents of 4 cells instead of just 1. Okay, okay, so that's the first polar body, and the secondary oocyte is produced after or as a result of meiosis 1. Okay, meiosis 2 is going to begin. Okay, it's going to begin right before ovulation. Okay, and then again, it's going to stop. Okay, so it gets to essentially the second step of meiosis 2. So metaphase 2, this is when all of the chromosome or the sister chromatids here are lined up on the metaphase plate. If and only if fertilization occurs, so sperm penetrates egg and deposits its own genome, is metaphase 2 going to be completed. And so it is possible that if a woman never gets pregnant, her oocytes will never actually finish the process of meiosis. Okay, so again, meiosis 2 is only completed if fertilization occurs. There's actually, the cells that are ovulated are not completed yet. Okay, so let's look at what ovulation looks like. So here we have an ovary. Here is structures called fimbriae, and this is the uterine tube, or more commonly called the fallopian tube. Essentially, what we're going to see, and we'll certainly go into this in more detail, but the oocyte is going to develop within its follicle. Okay, and it actually pushes against the surface of the ovary, and given a surge of hormones, okay, which we're going to get into, the surge of hormones is going to cause the follicle itself, which is this like moon-shaped structure here, to burst. And that bursting is going to essentially spit the oocyte, the egg, out of the ovary and into the space in the female's pelvic cavity. Okay, so I include this image because there actually is space between the ovary and the beginning of the fallopian tube. So ovulation is going to occur, it spits the egg out, and hopefully the egg makes it into the fallopian tube to ultimately go down into the uterus. And so one of the ways that kind of increases the chances of the egg actually getting to where it needs to go is the fact that there are these little finger-like structures at the end of the fallopian tube or the uterine tube to kind of create a current and more or less suck that egg into the uterine tube. And so those finger-like structures are called the fimbriae. It is possible for fertilization to occur out here. All right, so here the egg is not actually in the fallopian tube, it's not actually in the uterus. Instead, it's just in her pelvic cavity. And so it is possible, very rare, of course, and it's a good thing, that baby can be made outside the uterus and can actually implant and produce what's called an extrauterine pregnancy. Okay, obviously, natural childbirth is not an option when that occurs. Okay, so if everything works out right, the secondary oocyte, the egg, is going to be moved into the fallopian tube or uterine tubes as a result of these fimbriae. Unlike the sperm, the egg does not have a flagellum, and so it can't actually swim down into the uterus. Instead, there are cilia lining the uterine tube. Okay, and these cilia and peristalsis are going to slowly, over the course of almost a week, move that egg down into the uterus. Okay, we can see down here that sperm swim up and into the uterine tube. Usually, fertilization is going to occur within the uterine tube itself. Okay, so sperm joins with egg. Okay, their two genomes combine, therefore making a new diploid organism. This is the offspring here. As the cilia and muscular uterine tube push this developing offspring down into the uterus, cell division is occurring, and that is called cleavage. And we'll talk about that more in a later lesson, but essentially by the time this fertilized egg actually gets into the uterus, it has dozens of cells, and those cells are starting to differentiate into different types of tissue that will become essentially part of the placenta, will become the different tissue types of the developing baby, and some extra embryonic membranes, so essentially the membranes that are going to support baby, so surrounding it with amniotic fluid. All of those things develop literally within the first week after fertilization, or the precursors to it do anyway. Okay, if the secondary oocyte is not fertilized by a sperm, it never completes meiosis II, and it eventually is going to end up in the uterus. It does not implant into the endometrial lining. And 2 weeks later, right, so 1 week to get down into the uterus, 1 week within the uterus, then the secondary oocyte as well as the endometrial lining is going to be shed via a menstrual period. Okay, so let's briefly compare oogenesis and spermatogenesis. Both of these processes produce viable gametes, right? So haploid cells that are able to join with other haploid cells to ultimately form a new diploid genetically unique offspring. Both involve meiosis I and meiosis II, right? However, spermatogenesis ends with the production of 4 unique cells, and all of these are viable. All of these in theory, can fertilize an egg. Okay. In oogenesis, the other 3 copies of DNA are going to be essentially stored in these polar bodies, which are just like little storage areas. They're not actually doing anything for the egg or the female. They're ultimately going to degenerate and die, but they're essentially donation of all their cytoplasmic contents into one single cell is going to allow that cell to survive for several days on its way to the uterus. Okay, so before mom's blood supply can ultimately start feeding this developing baby, there needs to be a little bit of stored food already inside the cell, and that is provided by the 3 other cells that are pretty much eliminated right away in this process. Okay. Spermatogenesis begins at puberty, continues all the way until the man dies. Oogenesis begins before birth and pauses until puberty and then continues until menopause, so only a few decades really. Spermatogenesis is fairly accurate, so only about a 3 to 4% error rate as far as copying DNA is concerned. Oogenesis has a much higher error rate, and a lot of this is because of that play pause multiple times over the course of her life. Individual cells can be paused in prophase 1, just stuck there, all the chromosomes kind of wrapped up together, and then 40 years later, okay, now we have to like unravel them and pull them apart and continue with this process. And so there is a fairly high error rate, especially towards the end of the woman's reproductive years, because of that pausing. Now, a lot of the genetic mutations associated with the female error rate are like chromosome number abnormalities, right? So Down syndrome, for example, is having an extra chromosome. The division of the DNA did not proceed appropriately, and so the egg might end up with an extra chromosome. Okay, so that's some of the issues that you might see in a female. In males, the same stem cells, or genetically the same stem cells, are going to replicate over and over and over again for many, many decades. And so if a male fertilizes an egg in his 60s, 70s, 80s, his chances of producing an offspring with genetic abnormalities is greatly increased because of the so many times these cells have been copied. And yes, it's only a 3 to 4% error rate, but an error in copying DNA when he was 21 is still going to be there all the way when he is in his 80s, right? And not just that one when he was 21, but all of the other errors accumulate over the course of his life and can produce a fairly high risk in his older age. Okay. And again, to compare, spermatogenesis starts at puberty, continues throughout the life. Oogenesis begins before birth, pauses, and then ends at menopause. All right, so I want to show you a brief animation of what is happening in meiosis, and at the same time, what is occurring in the follicular development. So what is happening in the follicle? Before I hit play, I want to point out that this here is the oocyte. So this is the developing cell that will become 1 gamete and 3 kind of extrapolar bodies. And the cells that are surrounding this are called the follicle. So let's look at what happens at the same time ametogenesis is occurring.
A primordial follicle consists of an oocyte surrounded by a single layer of squamous granulosa cells. A primordial follicle becomes a primary follicle as the granulosa cells become enlarged and cuboidal. The primary follicle enlarges and the granulosa cells form more than one layer. The zona pellucida forms around the oocyte. The structure is called a secondary follicle when fluid-filled vesicles develop among the granulosa cells and a well-developed capsule, or theca, becomes apparent around the granulosa cells. The theca possesses internal and external layers. The structure is called a mature follicle when the fluid-filled vesicles form a single antrum. At full maturity, the oocyte is located in the cumulus mass. During ovulation, the oocyte is released from the follicle, along with some surrounding granulosa cells of the cumulus mass, called the corona radiata. Following ovulation, the granulosa cells divide rapidly and enlarge to form the yellow corpus luteum. When the corpus luteum degenerates, it forms the white corpus albicans.
Okay. So as we can see, the follicle changes quite considerably over the course of a single month. Over the course of a single developmental phase of the egg. And it's going to be really important in kind of the next phases even after ovulation. Okay, so let's take a look here. We're going to follow the follicular developments. Remember that meiosis is occurring at the same time inside this follicle. Okay, so the primordial follicles all kind of hanging out in what's called the egg nest. Okay, so this here is the ovary itself. On this end, the end of the ovary that's actually farthest from the fallopian tube is what's called an egg nest, and that's where mom's entire supply of eggs, of oocytes, is going to reside until those couple are selected and one is going to continue the process of meiosis every single month from puberty until menopause. Okay, so again, these are the primordial follicles, and this is the egg nest, right? How clever. So in the primordial follicle exists the inactive primary oocyte surrounded by that simple squamous layer. So one skinny little layer of flat cells. Okay. Once one of those primordial follicles is selected to continue, we're going to develop what is called the primary follicle. Okay, so the primary follicles, just as the video just showed, develop, or the name changes when those really flat single layer of cells are going to start getting a little bit fatter. Okay, so they're going to kind of blow up lots more cytoplasm within them, so they're going to look a lot more cuboidal instead of squamous. Okay. When the shape changes, these cells surrounding the oocyte are going to be called granulosa cells. Okay. The granulosa cells are separated from the oocyte by something called the zona pellucida, right? So zone, right, the area, pellucida, so lucid, clear, or translucent. So this is the translucent zone. Again, this is developing literally between those cuboidal cells called granulosa cells and the oocyte itself. Now, the primary follicle is also going to develop a second type of cell. Okay, so we get differentiation of the cells during this phase. Around the outermost part of the follicle is going to be a layer of what are called thecal cells. And as we're going to see, the thecal cells and the granulosa cells are going to be really important in detecting hormones in the HPG axis, as well as producing hormones, and of course, regulating the developing oocyte within. Right, just like the nurse cells or Sertoli cells in the testis are going to kind of nurse along the developing sperm, these granulosa cells and thecal cells are responsible for nursing along and regulating, guiding, growing the oocytes within. Okay, and of course, they're going to produce estrogen. Okay, so if we look over here, we can see the oocyte, the zona pellucida, and multiple layers of those cuboidal cells, which are called granulosa cells. By the end of this primary follicle stage, there's also going to be this thin layer of superficial cells called the theca. Okay. The secondary follicles are going to continue growing. So those granulosa cells are all going to be undergoing the process of mitosis, making more and more and more of themselves. So this allows the wall of the follicle to thicken, and it kind of exponentially increases the activities of those granulosa cells. What they're doing is actually secreting a type of fluid. This fluid can be filled with nutrients and different chemical mediators, but essentially they're secreting out fluid, and every time mitosis occurs, we kind of double the amount of fluid that is secreted. Okay, and so initially this fluid is secreted into kind of discrete little pockets in between the cells, but ultimately those pockets of fluid are going to converge, forming this larger fluid-filled cavity called the antrum. Okay, let's go back up here. We have been walking through the meiotic events of the oocyte. Here's the oocyte. And now we're switching gears into talking about what's happening in the follicle surrounding it. The primordial follicles started to develop into cuboidal cells. The cuboidal cells would ultimately differentiate into thecal and granulosa cells. The granulosa cells start to secrete lots of fluid, and those fluid-filled compartments are going to converge into one big, what's called antrum, or a fluid-filled pocket. Okay. This final follicle here, right, the one with the antrum, is called a lot of different things, right? You're used to this by now. A tertiary follicle, an antral follicle, a vesicular follicle, or a Graafian follicle. If you're reading about this stuff, you will see it written all of these different ways. Okay. And so at about day 10, after the primordial follicle and its internal primary oocyte were selected for development, the tertiary follicle is going to be complete. Okay. Once the secondary follicle becomes a tertiary follicle, it's roughly 15 millimeters in diameter. So 1.5 centimeters, we're talking like 2/3 of an inch, right? So you can actually see the follicle with the naked eye. Okay, the oocyte is going to be projected into the antrum, so it's actually surrounded on most sides by this fluid-filled pocket. Okay, the oocyte is surrounded by granulosa cells. Right, so we can see these nice little cuboidal cells. They are granulosa cells. There's also this kind of crown of granulosa cells around the oocyte itself. This is called the corona radiata. Corona, like crown, we're all very familiar with corona at this point. The surface proteins on the coronaviruses are like a crown, and so here the crown around the oocyte is called the corona radiata. Okay. Okay, so it is the tertiary or antral or vesicular or whatever you want to call it follicle that is responsible for producing ovulation. Okay, so the tertiary follicle is going to be triggered to burst, releasing not only the oocyte itself, but the corona radiata and the zona pellucida that is between those granulosa cells and the oocyte itself. Okay. In our next lesson together, we're going to be talking about different phases of the female cycle. There's a lot of different overlapping phases, and we can be talking about what's happening in the ovary versus what's happening in the uterus. And so all of these things are going to come together. For our purposes right now, right, before we get into all of this in our next lesson, is that ovulation marks the end of what's called a follicular phase. So the follicle is kind of calling all the shots. This is what the main structure is within the ovary, and the beginning of the luteal phase. Okay, and so the follicular phase is about the first 2 weeks. The luteal phase is 14 days, the 14 days after ovulation, right? And that one is consistent. Okay, and so the first 14 days are characterized by the follicle itself. So primordial all the way through tertiary, ovulation is going to occur based or as triggered by a huge spike in LH and to an extent FSH. And the follicle that's left behind in the ovary is going to become something called the corpus luteum. And so that is the beginning of the luteal phase because the corpus luteum is going to be regulating what happens next. Okay, so the tertiary follicle itself is going to become the corpus luteum. The granulosa cells of the follicle don't just stop doing things. Instead, they are going to continue actively dividing via mitosis, and also they are going to ramp up their production of hormones. Okay, so specifically progesterone and estrogens are made by the granulosa cells, and they are going to be secreted more and more and more over the next 14 days after ovulation. And so the idea here is that particularly progesterone is pro-gestation, pro-pregnancy. And so if fertilization was going to occur, it's going to be within the next 2 weeks, right? In fact, within the next day or two. And if that happens, the corpus luteum is actually responsible for maintaining that endometrium to sustain the developing embryo. Okay, so finally, let us look at all of these events happening simultaneously from pre-birth all the way to the end of the monthly cycles. All right, so before birth, stem cells are going to replicate, making lots of primary oocytes arrested in prophase 1. These primary oocytes are surrounded by a single layer of skinny little squamous cells called the primordial follicle. These primordial follicles with their primary oocytes are arrested in place. Nothing happens for the next many years. Okay, so each month from puberty through menopause, one of those primary oocytes is going to be selected, and it is going to continue meiosis, complete meiosis 1, and begin the process of meiosis 2. While meiosis is occurring in these cells that might have been stuck in place, frozen in time for decades, the follicle is going to develop as well. So the primary follicle is designated or characterized by squamous cells getting fatter into cuboidal cells. The cuboidal cells are going to replicate, making many and many, many layers. These are called granulosa cells. They are separated from the oocyte by the zona pellucida. Okay. Cells are going to differentiate into what are called fecal cells, and they are going to form a shell around the entire follicle. The granulosa cells are going to secrete lots of fluid, and those fluid-filled compartments are going to converge into one single antrum. And so right before ovulation, there is the classification, the development of this vesicular, antral, tertiary, Graafian follicle, where the oocyte itself is surrounded by the corona radiata, embedded within the fluid-filled antrum, and surrounded by both the granulosa and fecal cells. Now, for reasons we are going to get into in our next lesson together, luteinizing hormone, LH, is going to spike. Tons of it is released all at the same time, and this is going to trigger that follicle to burst. And essentially, the follicle is going to spit out the oocyte with the corona radiata and the zona pellucida out into the space in between the ovary and the fimbriae. The fimbriae kind of pull in that oocyte and hopefully create enough of a current so that the oocyte is then going to start going down the fallopian tube towards the uterus. Now, the follicle that's left behind doesn't just go away. It's going to be converted into the corpus luteum, which is going to continue secreting estrogen and progesterone throughout the next approximately 10 days, okay, unless there is a fertilized egg. Okay, we'll get into that here shortly. So the corpus luteum continues to maintain the endometrium and possibly the embedding or implanting fertilized egg. If there is no pregnancy, okay, so there was not fertilization of the egg this time, the follicle is going to degrade into something called a corpus albicans. Albicans means white, and so essentially this becomes a little scar within the ovary of the previous follicle. Okay, as the corpus luteum degrades, it's going to produce less and less estrogen and progesterone, and so without these high levels of hormones that are pro-gestation, the endometrium is no longer going to be maintained, and it is then going to be shed via a menstrual cycle. Okay. One final note here, meiosis 2 is not necessarily completed. The only way meiosis 2 is going to be completed within that oocyte, right, so this final division here, is if sperm fertilizes the egg. Now, the other 3 copies of DNA here are all stored in what are called polar bodies, and they are just they are just there so that we can get this very large, energetically equipped haploid gamete. Okay, so that is all for today. Thank you guys so much for stopping in and watching. This will be completed in our next lesson together.